Hello, I am Gözde Cilingir, a postdoctoral research fellow from the University of Zurich in Switzerland. Today, I will be talking about the first chromosome-level Aldabra giant tortoise genome assembly that we produced by using PacBio High-Fi sequencing and Hi-C scaffolding. The Aldabra giant tortoise is one of the two giant tortoise species left in the world, together with the Galapagos giant tortoises. The natural distribution of Aldabra giants is restricted to Aldabra Atoll in the Western Indian Ocean. Due to their extremely limited distribution in the wild and the threats posed by climate change, the species is listed as vulnerable on the IUCN Red List. Aldabra giants have been successfully used in several rewilding projects in the region to replace extinct endemic giant tortoises and resurrect degraded island ecosystems functionally. To reliably assess and ensure the genetic health of the wild and rewilded populations, we need high-quality genomic resources such as a reference genome, which is lacking for the species. To start building the reference genome, we first performed PacBio High-Fi sequencing with a blood sample from an individual named Hermania, who was brought to Zurich Zoo in 1955 from an unknown location within Aldabra Atoll. By using the KMR frequency of 21x Hi-Fi reads, we estimated the genome size of Aldabra giant tortoises as 2.4 gigabases and the heterozygosity rate for the genome as 0.072%. For the draft assembly, we used several different genome assemblers that were built for processing Hi-Fi reads. Of these softwares, Hi-Fi SM provided the longest counting N50, and we carried on the contamination scan with this version. Several different approaches were used for the contamination scan, but we did not detect any contamination signals. Then we mapped available short whole genome sequencing reads and removed contigs with odd levels of coverages. The contaminated draft assembly was coupled with approximately 85x chromatin conformation capture data, or Hi-C-seq data, for performing genome scaffolding. Overall, a total of 26 pseudochromosomes were anchored, corresponding to 98% of the estimated genome size, yielding a chromosome-level assembled reference genome with an N50 of 149 megabases and a BUSCO genome completeness of 97%. To put these genome quality metrics into context, we can refer to this table of all chromosome-level assembled turtle reference genomes with their quality metrics. As you can see, the Aldabra giant tortoise genome at the top has the longest contig and scaffold N50, making it the most contiguous chelonian or turtle reference genome available. Additionally, it has one of the highest levels of BUSCO genome completeness. After genome sequencing, assembly, and scaffolding, we generated a species-specific de novo repeat library to identify, classify, and mask repetitive elements in the Aldabra giant tortoise genome. As a result, 47% of the genome was detected as repetitive. For gene model prediction, we performed ab initio and evidence-based predictions. As evidence, we collected already available RNA-seq data produced from different organ types coming from closely related turtle species. We also produced around 1 gigabase of iso-seq data produced from Aldabra giant tortoise blood and utilized the orthologous proteins of all vertebrates. As a result, we predicted almost 24,000 genes in the genome, of which 94% were functionally annotated. Following the annotation, we investigated the collinearity of Aldabra giant tortoise chromosomes with 3 other chromosome-level turtle genome assemblies from 3 different turtle families. We analyzed the largest 10 chromosomes, corresponding to around 75% of the Aldabra giant tortoise genome assembly. Among genomes, we found that synthetic regions and rearrangements correspond to 89 to 94% and 0.8 to 3% of the compared genome portions, respectively. The high ratio of synthetic regions that we found is between turtle taxa that diverged around 50 to 70 million years ago and is in agreement with previous studies where the base substitution rate or evolutionary rate of turtles was found to be relatively low. To assess the utility of our reference genome resources to improve genomic monitoring and inform rewilding efforts, we performed low-coverage whole genome resequencing focused on rewilding within and among island genetic We performed low-coverage whole genome sequencing, focusing on revealing within and among island genetic differentiation within the Aldabra population, as well as assigning likely origins for zoo-housed individuals. We first assessed the global genetic structure of the samples using a principal component analysis. Based on a total of 6.6 million variant sites, individuals from Malabar and Grantaire split into two groups. Both those samples were grouped within the group of Grantaire individuals, revealing the most likely origin for these individuals captured in the 20th century. The principal component analysis also reveals a finer scale east-west population structure within islands, confirming our recent results based on the DRAD sequencing. If you're interested to read more about the study of ours, you can scan the QR code below. We also assessed genetic structure using unsupervised Bayesian clustering, which revealed results mainly agreeing with the PCA. The admixture analysis for K equals to 2 clusters revealed a main split with groups formed by East Grantaire together with East Malabar, opposed to West Malabar. South and West Grantaire individuals were assigned to both groups. At K equals to 4, each major sampling region was assigned to a single cluster. The zoo individual Malaika showed a genotype highly consistent with South and West Grantaire individuals. The individual Hermania was assigned to different Grantaire regions. Assessing the genetic health of a species is crucial for its long-term survival, and one major aspect of genetic health is mutation load. For a first glimpse at the distribution of putatively deleterious mutation in the Aldabra giant tortoise genomes, we performed a variant annotation analysis. The analysis predicts the effects of genetic variants and allows estimating the expected impact. The variant sites were identified via low-coverage whole genome resequences, as I explained in the previous slide. Of all the 7 million SNPs identified, we detected groups of SNPs with a putatively high impact on gene function, identified as loss-of-function variants, and to have nonsense-mediated RNA decay effect. In conclusion, of all 44 tortoise species, only 3 reference genomes represent the Testudinidae family, the family of tortoises or land-dwelling turtles. So the new Aldabra giant tortoise genome fills an important knowledge gap. In our study, we provided the first high-quality chromosome-level annotated genome for the Aldabra giant tortoise, and in fact, to the best of our knowledge, it's one of the best assembled turtle genomes available so far. Further, we also show that high-quality resources can be combined with low-coverage resequencing to gain insights into the genetic structure within the wild population of Aldabra giant tortoises, as well as to resolve the exact origin of zoo-housed individuals or ex-situ individuals. In the end, understanding levels of genomic diversity in both native and ex-situ populations is crucial to inform rewilding efforts and prioritize conservation efforts. Additionally, by utilizing the gene annotation for the Aldabra giant tortoise genome, we identified putatively deleterious mutations to be used in the genetic health assessment of the wild and rewilded populations. Finally, given the giant tortoise's exceptionally long lifespan and large body size, the high-quality genome will also inform comparative genomic studies focused on the genetic underpinnings of aging and gigantism. With that, on behalf of our team, I would like to thank all our collaborators and our funders. And of course, I'd like to thank you for your kind attention.